Hello Imbibers, a very warm welcome to all of you children. So today we are here to revise about the two important members of the biological classification, which is the monera. Of course, we're going to revise protista also. For the very first, we're going to revise about the monerans. Yes, so you know children, in the previous session we have already discussed individually about each and every part of the kingdom monera, but today we're going to basically only have this quick revision session or a one shot revision session about the monerans. Yes, we're going to discuss what are the different types of bacteria existing. Yes, as you know, in the sixth kingdom classification, the monerans were further divided into the archaebacteria and eubacteria. So we're going to discuss the features of those also. Yes, and we're going to have all the discussion related to the monerans. Yes, before that, let me introduce you to myself. I am Anam Khan. I'll be your biology expert for the session 2023 and 2024. And like we have been discussing all the important and essential topics related to biology and of course botany, we're going to basically discuss in future also about that. Yes, so without wasting any time, let's revise, let's start our session, yes, which is the kingdom moneras revision. So children, as you know, as we move forward, you know, the concept today would be totally related to the journal features and of course the types of bacteria. Every type of bacteria would be discussed in today's session. Yes, and children, talking about the very first thing, which is the kingdom monera. Let's see what are the basic features of the monerans, why there was a necessity, why there was a need to keep the monera in a separate kingdom. Yes, so the very first features or the very first features of the monerans, which basically meant to be kept in the different, they're not identical to the other organisms which are being kept in the kingdom protista, fungi, animalia, plantae are these. Yes, these are unicellular organisms. What are they? They are unicellular organisms. And what do you mean by unicellular organisms? Simple, unicellular organism means single cell. All the function, all the necessary function, or I would say the survival, everything, the digestion, the assimilation, whatever you have to do for the survival, it will be done within the single cell. So they are typically unicellular, single celled organisms. Yes, they have a ribosome. Ribosomes are needed for the protein synthesis. Yes, protein synthesis. So what kind of a ribosome they have? They have a 70S type of ribosome. Yes, they have this 70S type of ribosome. The other feature is that their DNA lacks nuclear membrane. Yes, their DNA, they lack nuclear membrane. Just a second children. Okay, their DNA lack nuclear membrane. It means a well-defined nucleus is not present in the monerans. Yes, they have mitochondria also, they have lysosomes, they have plastids, they have Golgi apparatus, and endoplasmic reticulum is absent. Yes, mitochondria, lysosome, plastid, Golgi body, ER is completely absent in the monerans. Yes, they are typically prokaryotic organisms, and when you say prokaryotes, you know we are calling about the organisms which have primitive nucleus and lack all the double membranal organelle. Yes, children, next, how do they reproduce? They have very simple way of reproduction. It is, they are being done by asexual method, by binary fission. Yes, through the budding also. Yes, so these are the two possible ways for them to reproduce. One is the budding, the other is the fission. Yes, or it is binary fission. Majorly, these are known as environmental decomposers. What are decomposers? Which feed on the dead and decaying part and basically mix the nutrients back into the soil by decomposing the living organisms. Yes, when they are dead, these feed on them, they basically consume all the nutrients and mix it back with the soil. Yes, so decomposition is I would say the method to decompose the dead organisms is a total responsibility of the monerans. And how do they obtain their nutrition? They obtain their nutrition through the autotrophic nutrition parasitic nutrition, heterotrophic and saprophytic. Autotrophics, you know, they're basically making their own food. They are heterotrophic, they might feed on the other organisms also. Yes, or they obtain their food from the other organisms. Next, they could be parasitic, which is completely dependent on the host body, and they could be saprophytic, which are dependent on the dead and decaying organism. Clear? So these are the few features of the monerans, the few unique features which makes them different from the other organism, which basically is created a necessity to have their own kingdom called kingdom monera. Let's see what we have more in the kingdom monera. Talking about it, next we have about the monerans is that the general more features. Let's see what are the more features they have. Yes, the most important features which are absolutely important is that they have a prokaryotic nature. This we have already discussed, that they do have a primitive nucleus. The nucleus is not that well developed. And next, they have membrane bound cell organelles completely absent. The mitochondria, the plastids, the ER, yes, they are completely absent. Yes, membrane bound organelle, membrane bound cell organelles are completely absent. Yes, non-membranous cell-bound organelles ribosome is basically present. Yes, there is a 70S type of ribosome. As you can see, there are two subunits of the ribosomes. They are basically used for the protein synthesis, and since they are also protein synthesized, they also synthesize protein. The 70S type of ribosome, which is non-membranous cell organelle, is present in the monerans. Yes, and they have cyclosis, which is completely absent, and they have single enveloped development or single enveloped system. It means none of the part of this whole organism will be there with the double membrane structure. The whole construction of the whole prokaryote is always of single membrane, single enveloped system is there in the prokaryotes. So these are few just general features of the monerans. Yes, moving forward, discussing more about the monera. Next we have the most important feature, which is absolutely very the talk of the town, which is the cell wall. What the cell wall is made up of the monerans. So talking about the monerans, the cell wall is made up of peptidoglycan. Exception is only the mycoplasma. The made up of peptidoglycan. They show both autotrophic and heterotrophic mode of nutrition. They are both can make their own food, and they are also obtaining their food from the other organisms. So they are showing their both heterotrophic and autotrophic mode of nutrition. The primary method of reproduction is completely asexual, that is what we have discussed, but there is a false sexual reproduction activity taking place in the prokaryote. That is what we call as parasexuality. Yes, that parasexuality is a unique method of sexual reproduction, which is not actually a sexual reproduction, but it's somewhat like asexual reproduction. So we call it as parasexuality. So parasexuality is being shown by them as a method of sexual reproduction. Yes, and they are cosmopolitan. What does mean you have cosmopolitan? They are basically present everywhere. They have a very simple cellular structure. So the more simple nature of an organism, the more I would say the more easy for them to exist anywhere, because the limitation comes with the complexity. Yes, since the bacteria, the monerans are very simple organisms, so they are typically cosmopolitan because of their simple nature. Their I would say their survivability in the adverse condition is anyway more than the other complex organisms. So yes, so children, they have nature being having simple cellular structure, and they have complex behavior. They might have simple structure, but they basically they also have some monerans also have the complex behavior, but they are typically present everywhere. They have this adjustment ability to survive in any kind of environment or the external conditions. Yes, So moving forward, talking more about the monerans, next we have about the cell wall. This which is the most important part of the monerans. What is the cell wall? If you see clearly, children, a cell wall is made up of peptidoglycan, just as we have generally already discussed about it. So children, the cell wall in bacteria is made up of peptidoglycan, and the peptidoglycan or murin is a polysaccharide consisting of amino acid that forms a mesh-like peptidoglycan layer outside the plasma membrane of most of the bacteria, forming the cell wall. So basically this peptidoglycan or murin, which is also a polysaccharide of amino acid, which a polysaccharide consisting of an amino acid, which is forming a mesh-like structure, outside the plasma membrane, which we call it as cell wall. So a typical peptidoglycan can be divided into two parts, as you can see, the peptide and glycan. Yes, peptidoglycan, it means, the peptide means what? The peptide will stand for amino acids, the proteins. Yes, and the glycan will be standing for carbohydrate. Yes, so basically peptidoglycan means the protein or amino acid plus carbohydrate create the cell wall of the monerans. Yes, so let's see what kind of amino acids and what kind of a carbohydrate exist in the monerans. We have peptide amino acid like L-alanine, D-alanine, T-glutamic acid and L-lysine. On the other hand, talking about the carbohydrate, we have a modified sugar called NAG and NAM. Please remember these two names, which is absolutely very important, and the question on this comes very often in the NEET examination also, that what kind of a carbohydrate is present in the cell wall of monerans. So they have N-acetyl glucosamine and they have N-acetyl murmuric acid. There are two particular glycan, two particular carbohydrate present in the cell wall of the monerans. Yes, please remember the cell wall constitution. It is very important for examination also, for the future attempt for the examination, NEET examination also, if you ever wanted to sit for your medical examination. So the composition of the cell wall is absolutely very important. Yes, we have discussed about the cell wall. Let's see what we have next, talking more about the monerans. Next we have the gram-positive and gram-negative bacteria. Yes, the gram-positive and gram-negative bacteria is one of the two types of bacteria. Major distinction between the gram-positive and gram-negative. So talking about the gram-positive and the gram-negative bacteria, the tetrapeptide chain joins with the pentapeptide chain, while in the gram-negative, tetrapeptide chain joins with the tetrapeptide chain. The only difference, as you can see, in the gram-positive, the tetrapeptide chain is joining with the pentapeptide chain, but in the gram-negative, tetrapeptide chain is joining with the tetrapeptide chain, another tetrapeptide chain. Here the linkage is indirect, here the linkage is completely direct. There is a difference between the thickness of the cell wall. Yes, there is a thickness between the cell wall. 150 to 200 angstrom is a thickness of gram-positive bacteria, and 70 to 120 angstrom is a thickness of gram-negative bacteria. Yes, the lipid content of the cell wall is very low. Yes, the lipid content in the bacteria which are gram-positive is absolutely low, while the bacteria which are coming under the gram-negative is having the cell wall content of lipid is 20 to 30%, which is very high. When you talk about the lipids, we are talking about the fat layer. Yes, fats, the lipids. So lipid content in the cell wall of the gram-negative bacteria is a very high, which is approximately 20 to 30%. So please do remember these two differences between the gram-positive and gram-negative bacteria. It is very important to know that these are the differences existing in both of them. Yes, Moving forward, discussing more about the monerans. Next we have the types of bacteria. Yes, we have types of bacteria on the basis of their shape, on the basis of their staining, and on the basis of the respiration. We have divided the bacteria on the shape. Staining, we have already studied the gram negative and the gram positive. Respiration method, they could respire aerobically, means they can respire in the presence of oxygen, and they can also survive in the absence of oxygen. So they could be anaerobic, they could be aerobic. Yes, and they exist in different shapes and sizes. So based on their shape also we have different kind of bacteria. So let's see, explore more about it. Now the shapes of bacteria existing, these are the shapes of bacteria existing. It could be cocci, bacillus, vibrios, spirillum, filament like, stalk like, budded. These are the few features of, these are the few shapes of the monerans. When we talk about the cocci, the bacteria is spherical or oval in shape. Talking about the bacilli, that bacteria could be a rod shaped with or without flagella. Yes, having a rod shaped. Next are the vibrios. They have shape like a comma or a kidney, like a comma or a kidney. I can show you just a second. Or a comma like a kidney. Yes, they have shape like this. Yes, so this is your comma shaped or vibrio shaped bacteria. Next we have the spirillum, which have spiral or coiled shape. Next we have filament. The body consists of small filaments like fungal mycelia. What is fungal mycelia? We have already talked about the mycelium, which is a filamentous like structure which is called hyphae. A network of hyphae will create a mycelium. So exactly the whole structure, the shape of this bacteria which appears like a filament is exactly similar to the fungal mycelia. Next, they can also be a stalk like. The bacteria possess a stalk, have a stalk like structure, and they could be budded. Budded, you know, the small birds. The body of the bacterium is having uneven budding at in the places. So these are the few shapes of bacteria existing: cocci, bacillus, vibrios, spirillum, filament like, stalked like, budded like. So they can exist in any shape and any kind of a structure. So these are the major shapes which are very common in the bacteria. Yes, moving on, talking about the monerans. Next we have the three major types, and the most important, very important, because these features basically led them to keep them in the. So talking about the three major bacteria, which is the halophiles, methanogens, thermoacidophiles. I know we have already discussed about this, but having a quick revision that what we have discussed about this is about the halophiles, methanogens and thermoacidophiles. So let's see one by one what we have to go through. Yes, the very first is the halophile. The name is already suggesting what would be the. They will be living in an extreme saline condition where the salt content is absolutely very high. You know it is not possible because the cell wall will burst. A normal living organism cannot survive in a very high salt condition, but this bacteria is living in an extremely salty environment. They have rich salt substance like salt ponds, salt beds and salt marshes. They are typically present on such places, and they are facultative anaerobes. Yes, they have facultative anaerobes. So children, they are the halophiles. The name halo means saline. Yes, living in the saline condition. They live in extremely salt environment. Yes, they are basically present in the salt marshes, so rich salt substrates like salt pans, salt beds and salt marshes, and they are typically facultative anaerobes. Yes, as you can see also, a particular bacteria is basically surviving in the salt. This is just a depiction, a typical depiction. Moving forward, talking about the halophiles more, how these basically able to survive in such a condition, what exactly led them to survive in such a extreme high salt condition. So the reason they can survive in extremely high salty condition is that it is a special pigment, bacteriorhodopsin. Please remember this. Please, that they have bacteriorhodopsin in plasma membrane which produces ATP from metabolic activity, not for CO2 fixation. Yes, so children, they have this special pigment in their plasma membrane which is called bacteriorhodopsin, which produces ATP from the metabolic, because ATP is necessary, no, for the survival. Energy is necessary for the functioning metabolic activities. So they produces ATP from the metabolic activities, not for CO2 fixation. Yes, it makes them stable in high salt concentration. Next, how they can survive in the extreme high condition of salt? They have mucilaginous covering, a slimy layer covering over it. Yes, they do not have a vacuole. Yes, the absence of vacuole nullifies, extremely nullifies the factor of what? The plasmolysis to take place extremely, or the hyper, to survive in the hypertonic condition. So they do not have a sap vacuole. Yes, and then they have internal high salt content. So it's not the external environment which is very salty, the internal environment also is extremely high with the salt concentration. And the example of this is the Halobacterium salinarium is one of the examples of an halophile. So if anybody asks ever that how the halophiles are surviving, you will say they have this pigment in their plasma membrane which is called bacteriorhodopsin, which does the generation of ATP from the metabolic activities. Rather than fixing the CO2, they have mucilaginous covering, they are having no sap vacuole, and they also have internal high salt concentration. Yes, and the example is Halobacterium salinarium. Okay, let's play this. Let's see what exactly it's going to show us. So children, as you see this, the cell wall, the plasma membrane, the light is coming, bacteriorhodopsin, ATP and metabolic activity. So these are the few reasons they are able to survive in such conditions. Next, talking about the other member of this, which is the methanogens. Very much aware of this, and you have also must have experienced also the products of the methanogens. As you know children, methanogens are strict anaerobes. They cannot survive in aerobic condition. They are extremely strict anaerobes surviving in the absence of oxygen. They are majorly present in the marshy areas. Yes, marshy areas, they are marshy areas, you know, the daldal areas. Okay, yes, they are where the soil is very dense, thick, like that areas. They will be present in such marshy areas. What they do? They convert formic acid and carbon dioxide. They convert formic acid and carbon dioxide into methane, which helps the help of the hydrogen. As you know children, methane is basically also used for various purposes. Yes, so what they do? They basically convert formic acid and carbon dioxide into methane with the help of the hydrogen. Yes, yes, and they produce methane as a byproduct of metabolism. Even they produce methane as a byproduct. All of you are familiar with the gobar gas. Yes, that gobar gas is nothing but methane. Yes, whenever you going to basically burn the cow dung, it is going to release that methane gas. So that is also called as gobar gas. So methane is also a byproduct of the metabolism of the methanogens. Yes, because they are also present. Talking more about the methanogens children, we have already talked about the methanogens. So basically they produce the gobar gas also. Next is the thermoacidophiles. So children, what are thermoacidophiles? Name is very clear. They are going to be present in an extremely high temperature. They can survive in the temperature up to 80 degrees Celsius. Can you imagine how the temperature? 30-40 mein to we started sweating so much. Yes, so 80 degree is basically the pH is low as 2 and the temperature is high as 80 degrees Celsius. Yes, they live in hot sulfur springs and geothermal vents. The example of thermoacidophiles are thermoplasma, Cephalobus, Thermoproteus, and the common thermophiles are the common thermophiles. So thermophiles are basically the bacteria which are present in the extremely high conditions. with the temperature and extremely low pH, which is absolutely very acidic. So that is why the name is thermo temperature acido low pH phials, living in extremely high temperature, low pH condition. Yes, so children, they live in hot springs and geothermal vents. This is how basically it looks like. This is where they will be present. See, the water is itself boiling because the temperature is so high. Yes. So children, moving forward, we have discussed about the 3 types of bacteria. Yes, the halophiles, the thermoacidophiles and the methanogens. Let's see what we have next to revise. We have the eubacteria, which is also known as the true bacteria. Yes, this is one of the bacteria which is extremely having all the necessary or special features, or whatever you can say, a true bacteria having 2 bacterial features, true bacterial features. They are also known as true bacteria. Once again, their cell wall will be made up of peptidoglycan, and they have pili in some bacterial cells that helps in the parasexuality, the presence of the pili. Yes children, the presence of the pili helps in the parasexual nature, or you can also say a sexual reproduction method. A pathogen can adhere to host with the help of pili. Yes, children, a particular pathogen can basically stick to the host body. Yes, if the method of nutrition is parasitic, you have to have a host body, and how to stick to the host body? You can go and stick to the host body through the presence of the pili. Yes, they are again divided into 2 categories, which is gram positive and gram negative. We have already discussed about this. So this is what is about the eubacteria, which is also called the true bacteria. The cell wall is made up of peptidoglycan, and they have pili on the surface for the parasexuality. Yes. So children, talking about more of the, let's see what we have in this. Okay, these are the stain. This is gram positive, this is gram negative. Yes, can you show this? This is not showing the presence of what? The presence of dye on the cell wall, while the gram positive, which are very okay, the basic difference between gram positive and gram negative is that gram positive are easily taking, the cell wall can easily be taking up the dye. Yes, and next the gram negative, which are not basically taking up the gram stain. Gram stain means the stain, basically what we always call it, a stain means mark or something. So the gram stain, which basically the gram positive is, means the particular bacteria which can take up that stain, while gram negative which do not take the gram stain. So basically the basic difference is all about the staining the cell wall. Yes, so this is about the eubacteria. Next we have the mycoplasma, which is absolutely very important. The mycoplasma having a pleomorphic structure. What is a pleomorphic structure, children? They change their shape. They have a pleomorphic organisms, they can change their shape. It is a bacterial genus that lacks cell wall and protects the cell membrane. Yes children, it is a bacterial genus that absolutely, in the starting only we have discussing about the cell wall. We mentioned that the mycoplasma lacks cell wall. So this is of the genus of the bacteria which completely lack cell membrane. Yes, and they can replicate through binary fission. They also divide and basically reproduce through the binary fission. Next, the cytoplasmic division result in the multinuclear filament development. Yes, the cytoplasmic division which results in the multinucleate condition. Yes, and due to the lack of cell wall, they have actually resistance to drugs. So basically the mycoplasma, which can cause diseases, which can cause infections, the reason they are very much resistant to certain drugs means life-saving drugs, is because they do not have cell wall. They lack cell wall. They are naturally resistant to drugs, and mycoplasma does not have nucleus and other cell membrane bound organelles. Yes, they do not have a nucleus, and they do not also have the other membrane bound nucleus. So this is about the mycoplasma, which is a unique different or unique feature, or I would not say feature, but the unique bacteria which have certain features. Yes, they do not have cell membrane, they do not have cell wall, they do not have cell nucleus. Yes, and children, they do not also have membrane bound organelles. Yes, yet they show the cytoplasmic division, multinucleate filament formation, and yes, they are naturally resistant to the life saving and drugs, and that is why they cause the infection. Yes, moving forward, children, talking more about the other, which is the cyanobacteria, which is also called the blue-green bacteria. This is also the first oxygenic bacteria, which is the photosynthesis autotrophic, have chlorophyll pigment just like green plants. They are typically like green plants. They have chlorophyll pigment in their body which is necessary for the photosynthesis. Yes, plankton found in the oceans and seas are cyanobacteria. Yes, the cyanobacteria is found in the oceans and seas, and they produces the major fraction of oxygen in the atmosphere. That is, I was always say that these are the first oxygenic bacteria which produces oxygen through the process of photosynthesis. Yes, they can basically reside alone in colonies. They can basically be in colonies. As you can see, this is how they basically form colonies. This is how they basically form colonies. Yes, and asexual reproduction is through the transverse binary fission. The asexual reproduction is through the transverse binary fission, and there are few specialized structures that play an important role in the reproduction. Yes, there are few, I would say that there are a few specialized structure which basically let them survive in any kind of a condition, and they let them reproduce also, is the including akinete, hormogonia, hormocystis, and the spores. Yes, these are the certain features in the bacteria which let them survive in the adverse condition, and that is the reason they have been surviving over all these millions of years, since they are the first oxygenic photosynthetic bacteria. This is about the cyanobacteria. Yes, moving forward, discussing more about the monerans, we have okay, so this is all about the monerans. We have discussed a lot. Next, we are going to basically the archaebacteria, the types, habitat, basically eubacteria, everything has been discussed in today's lecture. We have revised a lot. Yes, so children, as you know, before going, what we do, we basically do the quick go through to the Embibe app. This is your Embibe app. Children, what you have to log in through your phone number or email ID, go and basically click on this, select your board, it could be CBSE, and select your class, which is the 11th CBSE. Click on the respective language and press done. Tada, you will land on this page, and as you click on the home page button, you basically go for the Embibe live classes. All the classes would be scheduled over here will come, and if you have missed any session, you can always go for the past Embibe classes. You can learn through this app, you can practice, you can test, and you can always see almost how much you have achieved. The next, children, is another amazing app, which is the Embibe Lens app. Yes, Lens app basically gives you an ability for you to just scan the text. It will highlight all the keywords and still you see everything in the 3D. We are going to basically also revise the Protesta. The next session would be about the Protesta revision. Whatever we have studied about the Protesta would be revised. Yes, until then, keep registering yourself for the classes. Have this amazing experience. Go for the live, come for the live classes on the Embibe app. Until then, we meet again. Keep imbibing. Take care. Bye-bye.
Keep imbibing. We believe in you.